Hello everyone, thank you so much for tuning in again. So we're going to continue with these review videos on stuff I've been learning in microbiology. So this chapter will be about prokaryotic characteristics as well as the varieties of prokaryotes that we'll be looking at in the class as a whole. So this is going to be grouping them by their metabolisms instead of their phylum or classes or orders or families. Some textbooks they do present them by class, order, family, all those things, but in the one that the professor went over, he wants us to learn them by their type of metabolism. So that's what we're going to be focusing on. Alright, so let me pop into the slides quickly. I had them open earlier, but my computer isn't agreeing with me right now, so we're just going to get started off rip here. So first we have this scientist. This is again in that portion of the notes that we don't really need to pay attention to because it didn't have that icon that the doctor specifically uses for things that we actually do need to pay attention to. So this is just a tiny bit of history. There's a scientist named Van Niel. Or I spelt his name wrong actually. It's I-E-L. Sorry about that. And this is going to be 1897 to 1985. And he found the purple bacteria. The bacteria did use light. However, the bacteria used hydrogen sulfide in order to oxidize CO2. So they oxidize hydrogen sulfide during CO2 fixation, and they didn't need oxygen. And then these purple bacteria were similar to the photosynthetic reaction organisms. There's a tiny bit of history there. Alright, let's go into the diversity of prokaryotes now. Only about 6,000 of the estimated million species of prokaryotes are known. So a ton of them are still out there unknown to science, and there are about 950 genera of the species. Molecular techniques as well as bioinformatics and mapping out the genome of these organisms. This helps with the discovery and the characterization. We have these molecular means of identifying them now if we have any new species. We have 5 categories that we're going to be looking at. So we have the anaerobic chemotrophs. We also have the aerobic chemolithotrophs. Next we have the anoxygenic phototrophs. Then we have the aerobic chemoorganotrophs. And then finally, we're going to be looking at the oxygenic phototrophs. Right, so basically 5 classes in the metabolic diversity of bacteria that are going to be a focus in this class. So let's get started with the anaerobic chemotrophs. Specifically, we're going to be looking at the anaerobic chemotrophs, but lithotrophs, chemolithotrophs. So anaerobic chemolithotrophs. Alright, so this means that O2, or oxygen, isn't the terminal electron acceptor, or the T the terminal electron receptor. We have this anaerobe. Anaerobe means without oxygen. So they don't use the O2 for their reactions to create energy. However, chemolithotrope means that they use reduced inorganic compounds as the source of energy. These include, gonna have to highlight some of this stuff here. O2 isn't the terminal electron receptor. Some of these examples of inorganic compounds for the obtaining of energy, we can have bacteria that use hydrogen gas. An alternative electron acceptor would be something like CO2 or sulfur. And usually these are archaea. An example would be a methanogen. And methanogen, so we have methane and then gen. That means to make or to generate. So these will make CH4. Alright, let's save that piece of information into our back pocket, because now we're going to be looking at methanogens as a whole. So actually, I'm going to bring this over and move it down, since now we're going to be talking about methanogens. Methanogens are, again, archaea. They're going to oxidize H2 gas into ATP. Their terminal electron acceptor will be CO2, and the common equation would be for hydrogen gas, which is the energy source. Do you know what? Let's color code this. The energy source, let's always make it in what's something that has a ton of energy. Let's see, how about this green? Energy. And then, of course, we're going to have our terminal electron. Electron, it's like negative. So maybe a red. It's positive is usually green, so terminal electron acceptor. And then we will have, of course, our products at the very end. So our terminal electron acceptor is, again, CO2. And these will, of course, methanogen. We're going to have methane and 2H2O. Again, methanogen, methane, there's our methane that's produced. We have this high sensitivity to oxygen, since they do not have peroxidase. There's no way for these archaea to defend against the reactive oxygen species or these strong oxidizing agents that are produced as byproducts of common biosynthetic pathways that the cell uses. So they're very sensitive to O2 since they don't have those peroxidases to deal with it. They're found in sewage, swamps, digestive tracts even. Cows will produce about 10 cubic feet per day of methane, so cow intestines. This is, again, another habitat, and when they're in the cow intestine, they could produce about 10 cubic feet per day of methane. Some uses in sewage, so methanogens will, of course, produce CH4, and then this is also put through other vats and other collection sites where you have other microbes working away at solids in this sewage, as well as other things just filtering it out. And then when we get to a stage where we have this methane gas, we could use it to power electricity. Alrighty, let's go into our anaerobic chemoorganotrophs. Still going with the theme of chemotrophs, but this time these are going to be anaerobic chemoorganotrophs. So instead of inorganic compounds as the way to make energy, we're going to be oxidizing organic compounds here. And specifically, not red, sorry about that. We're going to be looking at their respiration first. So respiration. And again, we're going to oxy organic compounds to help produce energy, or ATP. And then an example would be glucose. So going to highlight that. That's an important point. Anaerobes will actually be using sulfur as the electron acceptor. So sulfur. Again, we have sulfur and sulfate reducing bacteria. And these will make hydrogen sulfide. Oops, sorry about that. Let me highlight the type of bacteria there. And these products, this hydrogen sulfide is actually corrosive. Sorry, I didn't mean to use the highlighter. So if you have these bacteria in metal pipes, they're going to do some damage. They're going to corrode that metal. They're also important in the sulfur cycle. Again, we've talked about nutrient cycles in ecology and their importance to distributing all those compounds for the usage in other reactions and metabolisms of other organisms. There's a genera in here called the sulfovibrio, and then that's just an important genus of sulfur or sulfate reducing bacteria. Again, if we have our little guide here, so we need something like an energy source and then our electron acceptor. So organic compounds. This is again energy source, and then we have our sulfur as the electron acceptor, and then we're going to have CO2 and H2S as the product. Again, and then energy. All right, let's move on to another part of chemoorganotrophs. So this is going to be their fermentation. So anaerobic chemoorgano fermentation. Fermentation is when you're going to be, again, not using oxygen as the terminal electron acceptor. You're going to be using an organic compound as the electron acceptor. Many anaerobic bacteria will be fermenting. Again, the organic will be the electron acceptor in this situation. It's much different than the respiration of aerobic species. So basically what will happen in general, we'll have glucose, and then that will become pyruvate. Remember, 2 pyruvate because they have 3 carbons each. And then this will become like an acid or an alcohol, something of that type. If we pull out our little cheat sheet, we're going to have the, again, energy, and then our electron acceptor, and then products. Here we're going to have glucose as the energy source, then pyruvate as the electron acceptor, and then of course we have our acid and alcohol in the product side. very important species or genera to look at when you're talking about the fermentation of anaerobic chemoorganotrophs would be Clostridium. And these are gram-positive, and they form endospores. They're actually known as terminal spores in this case of Clostridium. And basically they tolerate O2 when they're endospores. And so they could also survive periods of heat, drying, chemicals, and radiation. We've talked how tough these endospores are, so they're able to survive into those conditions that normally anaerobics cannot. And of course they'll germinate when conditions improve. Symptoms would be like flaccid paralysis, tetanus, botulism. You don't want to eat home canned goods because of the dangers of these Clostridium bacteria to survive. So I'm just going to list some of their symptoms too. So tetanus, botulism. We always talk about pink sauce here. That's a home canned good. You don't want to get botulism from something like that. So those spores are going to still be there. They're going to survive if they're not properly processed. And again, flaccid paralysis, not something fun you'll ever want. Here's a little piece of trivia that the professor gave us. So there's a Gerber family, not the baby food Gerber family, but there's a Gerber family, and like 12 people died because of a condiment, canned beans, that were unfortunately canned incorrectly because of the spores of Clostridium. So that's just a little interesting yet horrifying effect of not taking the precautions when dealing with an endospore species such as Clostridium. All right, let's go and talk about lactic acid bacteria. So again, these are still our anaerobic chemoorganotrophs, just another type. Right, so these can be they can be aerobic, but they only ferment. So we're going to make fermentation only our largest point there. And that's because they lack catalase. That's another enzyme that protects against the strong effects of oxygen species that are made when metabolism occurs. So they don't have that enzyme that breaks down those harmful oxygen-containing molecules into something safer. So that's why they can't probably use anything other than fermentation. Common species is, or I should say genus is, Streptococcus. And these are in the oral cavity. Usually they're normal there. They're in your mouth. They look like chains. There's some that are pathogens, especially beta hemolytic asparagines and Thermophilus. So Streptococcus thermophilus actually makes yogurt. And then, of course, Streptococcus, they're chains. Staphylococcus are grapes. So that's kind of a way to determine between those two genera. I'm going to highlight that as well. Okay, there we go. All right, let's talk about lactic acid bacteria and their fermentation. So we're going to continue here, but specifically with their fermentation. So we have lactobacillus. So these are found in the mouth, the vagina, and they're rod-shaped. So mouth China, and these are, again, they're also going to be tolerant in those lower pH areas. They're going to be able to live there and inhibit pathogens. Since there's that lower pH there, they're able to thrive. Some other pathogenic microbes cannot. They're also important in culinary since they are used to make cheese, which is pretty interesting. I did not know that. We also could talk about the Prokaryotic species. And these are gram-positive pleomorphics. And they produce, of course, propionic acid due to their genus name kind of sounds like propionic, so they might have to do something with it. This acid is used to make antibacterials, as well as Swiss cheese. And they're found in the intestine. Very interesting group of bacteria there. Okay. Let's see where we are. Alright, now we're going to go into anoxogenic phototrophs. So let's look at that word. So this means anoxogenic. That means they don't make O2, and light is the energy source. Right. Sensing some similarities between photosynthetic organisms and these bacteria. And there's a reason for that. These are some of the earliest photosynthetic organisms. They use hydrogen sulfide or organics for reducing power. Remember, we were talking about all those reducing power molecules in the metabolism slides, so those are really interesting. They're found, of course, in lakes, bogs, mud. And let's go and talk about the equation here. So again, we have energy source, electron acceptor, product. So energy source, and then electron acceptor, or electron source, I should say, 6CO2 as the energy, and 12H2S. We'll make a sugar, so carb, or like glucose, I'm guessing. And we also have sulfur and 6H2O. There's 12 of those, and then one of those sugars, C6H12O6. Alright, we're going to be going into some types of anoxogenic phototrophs now. So, of course, we have our purple bacteria, which we were talking about earlier. Oh, not red. Sorry about that. I keep forgetting I switched back. Purple bacteria are gram-negative. They're photosynthetic because of these apparatus in the cytoplasmic membrane. The CPM. And this has a ton of folds for surface area. Because remember, reactions love when there's a ton of surface area. Okay, so we also have purple sulfur bacteria. And these will actually have gas vesicles for them to adjust the light or control their depth. And they're going to store sulfur in granules. And these are going to be intracellular. Intracellular is a key point here because another type of purple bacteria will actually storm outside. They're going to use H2S as the reducing power. Alrighty, so now non-sulfur bacteria. Purple non-sulfur bacteria will again live in moist soils, bogs, and paddy fields. They're going to use organic molecules as the reducing power. Remember, it's set up here. Anoxygenic phototrophs will either use hydrogen sulfide or organics as the reducing power, while non-sulfur purple bacteria will be using organics as the source of electrons. No gas vesicles. And these are going to be outside granules. So non-sulfur, out. That's kind of a key there. So non-sulfur and then out. So non and out. All right, let's talk about green bacteria. All right, green bacteria are filamentous. And of course, they could also make multicellular arrangements. They have gliding motility. They could have chlorosomes. And they have a ton of diversity in their metabolisms. So they're going to be like these weird stringy things because they're filamentous. All right, let's talk about oxygenic phototrophs next. Did I use green yet? No, I have not. All right. All right, this will say, so again, anoxygenic meant it can't make oxygen, or it doesn't make oxygen. Let me get back here. Sorry about that. Yeah, so these ones are going to actually make oxygen, and they're still going to use light as the energy source. Let me do that a little bit neater. There we go. Right, so our main type will be our cyanobacteria. Cyanobacteria are some of the earliest oxygenic phototrophs. And they're around 3 billion years ago. That is a huge amount of time. My goodness. That's enough time for like all the McDonald's ice cream machines to start working again, you know? Or the next time that the newest Avatar sequel, it will be 3 billion years in the future. Who knows? You know, it's a very long time. All right, so they began producing oxygen at 3 billion years ago. Water was the source of their electrons. And they play an essential role as the primary producer. As an ecological standpoint, they harvest sunlight to convert CO2 into organics. Then, of course, their equation might look a tad familiar if you know anything about plants. It's going to be the carbon source, 6 CO2 plus 6 H2O, and of course light energy to make C6H12O6 plus 6 O2. This looks a little bit familiar. So again, we have our carbon source, we have our electron source, and yeah, leave it in the comments what that kind of reminds you of. I should say carbon source there, I think. Alright. We could have unicellular ones as rods, cossi, or spheres, or spiracles, actually, not spheres. Sorry about that. I'm trying to read my handwriting and I messed up here. Spirals, not spheres. They're technically spheres because they're cells, but no. Alright, we also have multicellular forms called trichomes, it says. I'm actually ahead of myself here, so we're going to hit a pause, and we're going to say that they are gram-negative, and some can actually convert N2 to ammonia. And this is called nitrogen fixation. So some of them are capable of nitrogen fixation. They're gram-negative, that's another thing to point out. Okay, so now we should be going into the structures of cyanobacteria. So again, unicellular, that point that we made about unicellularity will be going down here. And then, of course, we're going to also have these filamentous multicellular trichomes. And then these kind of look like strands or filaments. They're going to also have gas vesicles. Again, they want to be near the top for some light, so we're going to use those gas vesicles to float and create a buoyancy kind of in order for them to traverse depth in water. We have blooms of these cyanobacteria. And these are, of course, large accumulations. These are caused by hot weather. And the heat will actually lyse cells to create something called scum on the surface of a pond. And the chloroplasts actually evolved. So again, it's like Pokemon. The cyanobacteria would evolve into the chloroplast over time. Let me center this actually. There we go. And bloom is kind of a dictionary definition here, or a key term, so we're going to highlight that as well. Okay, so now we have to go into the study of aerobic chemolithotrophs. Alright, so this means they do like oxygen. So we're going to put a little smiley face, and they're going to use inorganic for the source of energy. I'm going to highlight it too. Okay, so in this group we have sulfur oxidizing bacteria. Sulfur is the energy source, while H2S will also be another thing that they could use. Energy from oxidation of sulfur compounds, such as H2S or thiosulfate. H2S. I'm going to just put it underneath there. Alright, again, sulfur cycle. These will help in that recycling of this nutrient for other uses. We have filamentous, as well as unicellular lifestyles. The equation for this type of bacteria, or this category I should say, again, we'll have our energy source. We'll have the electron source, or in this case a terminal electron acceptor, so TEA. And then of course our products will be on that side. Okay, so first are the energy source and the terminal electron acceptor. Let me draw my boxes. Okay, so sulfur energy source. 1 1/2 of O2, so oxygen again, terminal electron acceptor. Aerobic means we're going to be using oxygen as a terminal electron acceptor. We're also going to have to add water. And then product is H2SO4. You know that one rhyme about Johnny where he drank something that's clear in a glass and he thought it was H2O, but it was H2SO4. That is sulfuric acid and it's highly dangerous. Right, let's talk about filamentous. Filamentous sulfur oxidizers, or FSOs, I guess. FSOs. These ones are going to be using intracellular granules for sulfur. They're going to be in sulfur springs, sewage, surface of sediments in marine and freshwater habitats. We have a species here called the, or a genus here called the Bagiatoa, and these ones are going to have gliding motility. We're going to also have some ones that do not move at all, such as the Thiotrix, and these can't move, they're immovable. And the interesting ones about these, the progeny, so we have something like a stick like this, and we have these other filaments coming off. Those filaments will actually detach, and they're going to move around via gliding motility. That's how those ones bud. They kind of have these filaments that will just swim off, you know. Alright, we could also talk about the unicellular sulfur oxidizers. A lot of different types of bacteria here. We have something called the, let me find my place. These are going to be oxidizing metal sulfides. A species or a genus that we'll be looking at is Acidithobacillus, Theobacillus actually. And of course these are going to be in terrestrial as well as aquatic habitats. Since they can oxidize metal sulfides, this is used for bioleaching. For example, so oxidation of gold sulfide will make H2SO4, and this will have a lower pH, and then the gold will be able to be soluble. That's what bioleaching is. We could also oxidize sulfur into fuels to sulfate. And removing sulfate will equal less acid rain. Ain't that nice? It's a good thing. We need to remove that sulfate. And acid runoff from acid rain, of course, could be as low as pH of 1, so extremely dangerous and corrosive. In our aerobic chemolithotropes, we also have nitrifiers. These are going to be gram-negative, and they're going to oxidize inorganic nitrogen. And that's going to be, of course, used for energy. There's a concern to farmers about ammonia leaking. or ammonium nitrogen should be more exact. And the problem is the ammonium nitrogen can deplete water. If O2, if the wastes are high in ammonium. Oh, I wrote that backwards. Sorry about that. NH3, not HN3. Two groups, so they usually grow in close association here. We have these working in tandem. So we have this first group, the ammonium oxidizers. So these are like nitrosomes. That's just an example of one. This is a type of a nitrifier, so their equation will be NH4, that's ammonium, plus 1 and 1/2 O2. This is going to make NO2 minus H2O and 2 hydrogen plus. Alright, so the most important thing here is that pay attention to that, keep that in your back pocket. Alright, so again, terminal electron acceptor is O2, energy source is ammonium. I'm going to just draw that down here too. Oh, there we go. So we're going to have kind of a mix and match here. Okay, so we have that equation again. Energy source, terminal electron acceptor, product of NO2 minus. Okay, so nitrite oxidizers will be this equation here, and that's going to be using that NO2 minus from this step. So this is actually going to go here. NO2 minus will be the energy source, plus again O2 1 and 1/2 O2 as the electron acceptor will make NO3 minus as the product. Nitrite oxidizers include Nitrobacter or Nitrococcus. Those are just two genus of those. We also have hydrogen oxidizing bacteria. These are pretty interesting. In our study of aerobic chemolithotrophs. Alright, so some types of these include Aquifex, Hydrogenobacter, and they're going to be obligate chemolithotrophs. They're going to be thermophilic. They really like those high temperatures. They're going to be in hot springs. These are going to be some of the earliest bacteria who exists on Earth. This is believed since they have low O2 requirements, and they're possibly available in certain niches. Due to the photochemical processes that split water. So, because of the splitting water. Alright, so again, energy source is H2. Terminal electron acceptor on aerobic species will always be oxygen. And that's going to make H2O. Going to, again, do our little boxes here. Alrighty. We have a few more left. We have the aerobic organotrophs next. So again, we're going to be using oxygen as the terminal electron acceptor. Now, our energy source will be these organic compounds. Alright, let's check out these species. I think I already used blue, didn't I? Let's use orange. This one I will be just underlining it instead of highlighting it so it's easier to see. Again, aerobic oxygen is the terminal electron acceptor. Chemoorganotrophs, they're going to be using organic compounds as a source of energy. And we have the equation here, organics. That's the energy source. O2 is the electron acceptor. And we're going to be making CO2 plus H2O as our products. It's going to do that for right now. Alrighty. Some inhabit specific environments. Others are just like everywhere. It doesn't really matter. Alright, so now we have some obligate aerobes. Again, obligate means they must be in an oxygenated environment since they're obligated to that oxygen. So oxygen requiring no matter what aerobes because they're going to be using that as the electron acceptor. We have genera here like Micrococcus. These are going to be gram-positive. They are found in soil, dust particles, inanimate objects, skin. They do have pigmented colonies. And they like dry and salty environments. That's not their flavor, that's just where they live. They like dry and salty environments. Not Micrococcus taste dry or salty. They just like those environments. Alright, Mycobacterium. These are the species that do not have the peptidoglycan in their cell wall. They have those acid-fast bacterial cell walls of mycolic acid. And then, of course, these are the acid-fast bacteria for that technique. We have pleomorphic rods of them. Notable pathogens of this genus would be tuberculosis bacteria, as well as the leprosy bacteria. Very dangerous stuff. Those are our obligate aerobes. We have more obligate aerobes here. We also have the Pseudomonas genus. These are going to be gram-negative. They're going to be rods. They have oxidase. So oxidase positive. They're going to be green or blue. They can't ferment anything. They're very strict aerobes. So Pseudomonas, wait, let me see. They could use degradation, and this is from plasmids. An important species of this widespread genus. Most of them are harmless, but some of them are pathogens, such as Pseudomonas aeruginosa, which we've used a ton in lab already. And they're very opportunistic. These are opportunistic pathogens. So you shouldn't bring flowers to a burn unit because you don't want the flowers or the soil that may be on those flowers to get into the patient's skin, and bam, you have a pathogen of the aeruginosa infecting them. You don't want that. Cystic fibrosis is also something caused by Pseudomonas. we can't make mucus, and so the Pseudomonas will stay in that area that is infected. We also have another genus called Thermus. And this is going to sound similar because this is where we get the Thermus aquaticus, or the Taq polymerase. So these are going to be gram-positive. They're thermophilic. They really love high temperatures. Again, the Taq polymerase for PCR comes from this Thermus aquaticus organism. And we need those heat-stable enzymes, since in PCR we're going to have 95 degrees Celsius to denature the DNA. Then we're going to have 55 degrees Celsius, a cooling period, to add the primers. And then we're going to kick that heat up again in the cycle to 72 degrees Celsius. And this is where the polymerase that is heat tolerant, that polymerase is going to start amplifying that gene of interest. Hopefully we'll make copies of it. I also have another genus here called Deinococcus. What's important about Deinococcus, they're radiation resistant. So they're kind of like the Hulk. They could resist all types of radiation. Dosage of radiation will shatter genomes. But then they have these enzymes that repair the damage, which is very, very interesting and pretty unique too, and as well as surprising. All right, let's talk about facultative anaerobes. These are, again, aerobic organotrophs. Facultative, again, means they prefer aerobic respiration if oxygen is available, but they could also ferment if needed. Okay, here we have the Corynebacterium. These are, again, those blue palisade-looking organisms. They're going to be gram-positive. They're going to, of course, have palisade arrangement, which means they're parallel. They kind of look like this. They have these, like, character-like appearance. And, of course, they're going to cause diphtheria, another disease. We also have these facultative anaerobes. Another group of them would be the enteric bacteria, or enterobacteria. Either way, it's a family of bacteria. These are going to be gram-negative, and they're going to be in the intestine, and they like to ferment glucose. Let me clear this up. There we go, enteric. They're found, again, in the intestines normally, but they could also cause typhoid fever, TYP. Sorry about that. And of course, this is caused by the Salmonella enterica. And there's a specific serotype. This is typhi serotype. We also have the bubonic plague, caused by an enteric bacteria. Again, a facultative anaerobe. This is caused by Yersinia pestis. Again, maybe pestis from the rats. Those are pests, so it hops on the fleas from the rat. Then you got bubonic plague, most of the world dying from this horrible disease in the Middle Ages, and that was long ages ago. Still around today in small pockets, mostly contained now. Thank goodness for the development of modern medicine as well as the study of disease. You still see bubonic plague pop up every now and then in remote areas, but thankfully we have ways of combating it. We also have the coliforms. These bacteria will ferment lactose and make acid and gas. Coliforms include E. coli, that famous species of bacteria that unfortunately sometimes ends up in food, and then you get contamination and recalls and stuff like that. Alrighty, that's basically the diversity of prokaryotic metabolic systems in these different types of bacteria. We also looked at archaea too, which was pretty interesting at the beginning. Again, to review, we have those 5 categories that we went over. We have, let me find them, anaerobic chemotrophs, aerobic chemolithotropes, anoxygenic phototrophs, aerobic chemoorganotrophs, and then oxygenic phototrophs. We didn't go in that order. We kind of ended with aerobic chemoorganotrophs, but we looked at these 5 categories, and yeah, a lot of interesting diversity happening there. All right, thanks so much for tuning in. Please do something nice for someone, and have a good one. Take care.